So it begins with multiple choice, about 70 questions on the Scantron. The exam's going to start with our last lecture about the digestive system. And remember, for all the digestive enzymes, I made a table on the board. You need to know the name of the enzyme. You need to know the product and the substrate and product. So if I say what enzyme breaks down maltose, you have to say maltase. Now, another possible choice could be disaccharide. It's a term I used in the lecture. It means any disaccharide. So I could break down sucrose, maltose, or lactose. If I talk about what enzyme produces amino acids, that would be the protease, and so on. So know the enzymes. Remember, the role of salivary amylase is more to make starch taste sweet, so we get more energy rather than any other reason. I didn't really spell that out in the notes, but I did tell the story in lecture, so if you hear it now, I guess it should be good. Okay, remember when we use bile to help make fats more water soluble, that's called emulsification. Remember what mechanical digestion is. That's any time we do things like chew food and then to break it into smaller bits. And then also chemical digestion, which actually begins in the mouth as well, is using enzymes to break down carbs or proteins or lipids or even DNA and RNA. Remember the role of acid, destroying bacteria in our food, at least some of them. Remember which part of the system does most of the absorption. Remember for the epithelial cell transport, what we're going to talk about is, first of all, so we had that image on the screen of the food on the food side, and then we had the wall, the epithelial cells. I talked about how to secondary active transport to get you from the food space, the lumen of the small intestine, into these cells. And then from the cells to the blood was facilitated diffusion. That's how I'm going to ask the question. So when I talk about going from the intestinal epithelia, that's the name of those cells, to the blood, that's facilitated diffusion. When I talk about going from the lumen of the small intestine into the intestinal epithelia, and these are the words I'll be using, that's going to be secondary active transport. Remember the role of intrinsic factor to help us absorb vitamin B12, not any other vitamin. We also talked about circulatory systems and such, and so remember the major blood cells, the erythrocytes, going to carry oxygen and help carry CO2. Leukocytes are going to help us fight infection. Thrombocytes, otherwise known as platelets, are going to help blood to clot. Now, if an animal doesn't have a circulatory system at all, it typically has cells close enough to the surface that it doesn't need to move stuff deep inside, like the cnidarian. There are only 2 layers of cells, so they're able to get all the oxygen they need from the outside world. Doesn't have to diffuse very deep. It's only when you have cells deep from the surface that you need some sort of circulatory system. When I say something about keeping cells so close to the surface, this is how most animals without circulatory systems get by. Remember that closed is more efficient than open circulatory system. Remember which groups from your chart have an open and closed circulatory system. For the mollusks, there's both, depending on which type of mollusk. The cephalopods have a closed system, the rest have an open system. Remember how stuff enters and leaves the blood? It's always through the walls of capillaries, not through any larger vessels or structures. And remember what filtration is: blood pressure, pushing water in small solids across the walls of capillaries. And then to get that filtered fluid back, we use the lymphatic system to put it back in the blood. Remember, veins have valves which help our skeletal muscle pump blood back to the heart. Remember, fish have a single loop circulation, and all the other vertebrates have a double loop. Remember, the electrical activity in the heart begins at the SA node and goes over the top two chambers and then the bottom two. If I said something about it going over the right side, then the left side, that's false. It's the two atria, then the ventricles. It's not the right side and then the left side. Okay, remember that hemoglobin uses iron, hemocyanin, which is what mollusks, cephalopod mollusks use instead of hemoglobin, uses copper. So remember those two elements, copper and iron, for blood transporting or oxygen transporting proteins in the blood. Remember the circuit of flow was sort of the last thing in those notes, how the blood flows from the vena cava through all the chambers and valves, and then to the lungs and back, and chambers and valves again, and then out to the body. So I'll list those things in order and see if you can pick and in the wrong order sometimes, and in the right order sometimes. Remember the pulmonary artery, pulmonary vein. Pulmonary artery carries oxygen-poor blood, even though it's an artery, which is unusual. Vein, pulmonary vein carries oxygen-rich blood, which is also unusual for a vein. Remember the tissue layers that are, so now I'm going to start getting into chart questions. Tissue layers that are found in cnidarians, the two that are found and the one that's not. Remember no tissues in sponges, and remember all the rest have three tissue layers. Remember the only acoelomate group is the Platyhelminthes, has three tissue layers with no coelom. So only pseudocoelomate group is Nematoda. They have three tissue layers and without a true coelom, a pseudocoelom. Remember the complete digestive system and which groups have that. Remember especially the annelids have segmentation as well as the arthropods. Remember the pseudocoelom is not completely lined with mesoderm. Remember the unifying characteristic of mammals, all animals, has something to do with the molecules on the surface of the cells. Remember the parts of the creatures we talked about that had calcium carbonate, and then which things had chitin. And I'll ask a couple questions about those things. Remember that cephalization, which is found in bilateral symmetry, is where you have the information processing and sense organs at the... remember the basic characteristics of all the phyla, like the arthropods and their exoskeleton, jointed appendages, and going on, the annelid segmentation and all that stuff. Remember the protists that live with the coral and give them food in return for a place to live. The type of protist those are. The food that the protists give them is carbohydrates made by photosynthesis, and which protists they are. Okay, tracheal system, remember the role of that. Now, we covered that in test 2, but we're going to cover it again here because we talked about it with Phylum Arthropoda. Okay, remember cnidarians without mesoderm don't have true muscle, so they can't handle prey that's very strong. They have to poison them to weaken them. Remember the difference between a coelom and a pseudocoelom. Remember what flame cells are, what Malpighian tubules are, what metanephridia are, what they're organs for, and which groups has which. Okay, remember which groups are the classic protostomes. That's arthropods, annelids, and mollusks. Remember, like I already said, bilateral symmetry favors cephalization. Remember that protostomes have the ventral nerve cord. Deuterostomes have a dorsal nerve cord. Remember, radial symmetry is used by animals that don't move very much. Okay, remember the arthropod exoskeleton has advantages like armor protection, a good place for muscles to attach for movement, prevents water loss, but a disadvantage is that it leaves the animal vulnerable during molting, easy to be hurt. Remember the foot is the tentacles in something like an octopus. Remember what amoebocytes do. Remember the structures, the three basic parts of a mollusk and what they are. That's the foot, the mantle, and the visceral mass. And the mantle, of course, secretes the shell. And the foot's important for movement. The visceral mass are just organs. Let's see here. Remember which group uses a GVC, which groups use a gastrovascular cavity to transport things around. It's both groups that have one. Remember which groups use which group uses its fluid of its pseudocoelom for transport, and which group uses the fluid of its true coelom for transport. Okay. Okay, remember the key to the tracheal system is even with your mediocre open circulatory system, you can still have a high metabolism. So that's the advantage that you get from the tracheal system. But remember, spiders use, all arachnids use book lungs, and the crustaceans use gills. Remember that even though snails live on land or are terrestrial, they actually use a gill for gas exchange. It's in a sac of water. We discussed that during lecture. Okay, remember another thing we talked about, another chemical we talked about recently was cellulose and how it's not digestible by us. It's dietary fiber in us, but some animals can get glucose from it if they have the right microorganisms living inside them. Remember, we store our excess glucose as glycogen, not starch. Remember that spongin is the protein part of peripheran's inner structure. And that the spicules are made of either silica or calcium carbonate. Remember, of course, the shell of clams, coral, the hard parts of a lobster shell, they all have calcium carbonate in them. Remember which protists are ancestors of animals are closest to them, and that would be the choanoflagellates. Remember, the reason jellyfish swim is to move food towards their mouth so they can capture it and eat it. Remember, I didn't really dig into this with you guys, but remember that I kind of skirted around it, but I didn't tell you about ulcers. Ulcers are when the digestive juices of the stomach, for example, are going to damage the lining of the stomach more than the cells can repair. The cells will divide to replace damaged cells. But if those cells are infected with certain bacteria, they don't divide fast enough, and the stomach's gastric juices wear a hole in the side of the stomach, which is called an ulcer. The harsh environment of the stomach then kills these epithelial cells faster than they can be replaced. Remember, the microvilli and villi increase the surface area of absorption in the small intestine. All right, the written part now is 50 questions, so 70 multiple choice true, false, and then 50 points for written questions. Most of that is going to be 6 families of questions. So I'm going to have question number 1. And then there's one answer for that. And it's the name of a phylum every time, all 6 times. And then the questions A, B, C, D, and F under number 1 are going to be about that phylum. So if I say a group has, let me just make something up here. It has a water vascular system. Hopefully you'll remember that's the Echinodermata. You'll write that in the first line. You got to spell it correctly. Including capitalization. All the phylum names are spelled on the test somewhere, so you shouldn't have much trouble spelling it correctly. Then the following questions will all be about that phylum. So then I might ask, how do they move tube feet, something like that. But if you put the wrong phylum at the top just so that you can answer, because you don't know the phylum I'm asking about, and you put the wrong phylum in and answer the questions for that won't get you anywhere. You could put the same phylum for every answer and answer the questions correctly for that phylum and not really learn anything. If you put the wrong phylum in the first question and you answer correctly for the right phylum, I'm going to give you credit for that. So, but each beginning question of those 7 question sections is meant to lead you toward one particular phylum, and then all the questions fit that phylum. If you get down the page a little bit and you say, hey, this doesn't seem to fit, this is something from arthropods, and I put this was phylum Annelida, then you better go back and rethink which phylum you're really thinking about. There would be certain, anyway. So that's how this is going to be, and they're mostly based on the chart. Things like respiratory surfaces, digestive system, circulatory system, all that stuff from the chart. Then there's occasional other questions, also including excretory structures, which they were on the chart for some groups. Had you put those in the remarks box. But there occasionally be other things like what chemical makes up the exoskeleton or something like that. Or if I ask about sponges, I might ask if they have tissues or something like that. Digestion in those guys is intracellular, and I may ask about that. Choanocytes for feeding. Amoebocytes are going to distribute food around the animal's body. Okay, you're going to need to know the symmetry. You're going to need to know which groups are capable of learning. And then some details like for sponges, no, the mesohyl is the jelly-like layer between the two layers of living cells. And then in cnidarians, the same sort of thing is called the mesoglia. So they're both going to be on the test probably, but you'll have to not mix them up. Remember which type of protists do the photosynthesis with coral. It's dinoflagellates. If you see anything else, that's wrong. Remember for body plans for cnidarians, that's polyp and medusa. Anemones are only polyps, never medusae. But hydras and anemones and their cousins corals, they're all polyps most of the time. The hydras go through a medusa stage, but it's much shorter. So hydras mostly use polyp. Anemones and corals only use polyp as their body plan, never medusa. Remember that in phylum Arthropoda, arthropods are the best adapted to life on land, by far, of any invertebrate. I've got people putting earthworms and all sorts of other stuff, but if you can live in a desert, and many arthropods do, then you're well adapted to life on land, and they're the only invertebrates that can live in a desert long term. And remember which group within Arthropoda, and you can use a common name, it doesn't have to be any scientific name, but which group within Arthropoda has the most species of any group of all of life. Okay, remember what chitin is, complex carbohydrate. Know all the respiratory surfaces for the arthropods. If I say a group is protostome, that doesn't help much, but if I say a group is deuterostome, there can only be one answer. We haven't covered chordates yet. So if I say what group has deuterostomes, it's got to be echinoderms. And then the adults have radial symmetry, the young have bilateral symmetry, that these can learn. They use their water vascular system to help them catch prey and move. They have a complete digestive system, their skin, gills, respiratory surface, and they use their coelom fluid to transport materials. And remember, nematodes, the only group of the pseudocoelom, they use their pseudocoelom fluid to transport materials. Remember, the cuticle is not there to protect them from desiccation. They can't live in dry environments. It's there to protect them from things like digestive juices, harsh conditions, but not drying out. So they have to live in wet environments. Respiratory surface, body surface for the nematodes, I guess that's obvious. Okay, one of the three cross sections, I'll give you a break and tell you it's not going to be the acoelomate one. It's either going to be pseudocoelom or coelom from the drawings I did. And I'll tell you to label all three tissue layers, label the gut or GDC, and then label the coelom or pseudocoelom. Then remember what's gastro about a gastrovascular cavity and what is vascular about a gastrovascular cavity. That is, gastro is digestion, vascular is transport.